So it's a real pleasure to be here this morning. Most of the work I'm going to talk about today I wasn't actually even involved in, so I'm very grateful that they're allowing me to talk about this work. And so it's a really interesting project where we've been developing a nucleotide analog for the treatment of Ebola infection. And the name of this compound is GS-5734. So I'd like to just get started with this map of previous filovirus outbreaks. So most people will be familiar with the West African epidemic, where there was high mortality rate, and also pretty much because of the scale of this epidemic, and it kind of crossed the globe. But interestingly, and something that was kind of a surprise to me was that epidemics occur, or kind of small outbreaks, occur of filoviruses across many different African countries every couple of years. But these outbreaks are generally within kind of remote areas, so they're contained. But I think the West African outbreak that happened in 2014, 2016, happened in a more densely populated area, and that was the outcome was this large-scale epidemic. So since that epidemic, there's been many companies that have been involved in trying to develop therapeutics and vaccines against Ebola, and so Gilead's one of those companies. So then the next slide, I just wanted to give a little bit of background on the disease course of Ebola in people. So like most viral infections, there's an incubation period. So after exposure, generally there's an incubation period of up to 4 to 9 days before people actually start to show symptoms. It has been reported that this incubation can last as much as up to 21 days, but that's generally not the rule of thumb. And then once people start showing symptoms, early symptoms are kind of indicative of any viral infection, so it's very difficult to differentiate between Ebola and other viral infections such as Lassa, which is kind of endemic to those areas where you're going to see Ebola. Also malaria as well. It's thought that some of these first cases were confused with malaria earlier on. And so then after a couple of days of showing those flu-like symptoms, that's when patients start to develop more serious symptoms, gastrointestinal issues, headaches, anemia. And then for those infected individuals that are not going to survive the infection, they pretty rapidly start to show hemorrhaging and coma and die around day 10 post-symptom onset. And so it's thought that people become infected with Ebola through a mucosal exposure, and where resident macrophages and dendritic cells become infected with the virus and transport the virus to the draining lymph nodes, where the virus then has access to the systemic circulation. So as I mentioned, there's a number of companies that are interested in developing therapies. The current standard of care, there is nothing approved. The current standard of care is supportive treatment. And in some of the Ebola treatment units that were established during the West African outbreak, actually some of those patients, if they were lucky, got up to like 21 different types of supportive care interventions. And so one of the things what we were able to achieve in that last outbreak was some efficacy with a ring vaccination strategy using a VSV-based vaccine that's currently being developed by Merck. But obviously that vaccine is not going to help individuals that are already really infected with Ebola. It will just protect close contacts. So there was a number of therapies, ZMapp, and so maybe a lot of people have heard of this particular therapy. It's a combination of 3 antibodies, and is administered to patients intravenously, and they were the only company that were able to perform a controlled randomized clinical study during the last outbreak. They didn't actually, they did see a 40% reduction in mortality in treated patients, but they didn't meet their endpoint just simply because of the fact that they didn't enroll enough patients. Favipiravir and TKM Ebola were 2 products that were tested, not in a randomized controlled study, but they were found not to be effective. There's also a lot of kind of contradictory data around on whether or not convalescent plasma or convalescent whole blood is actually effective. So this picture here is in the slide because the ZMapp antibodies are actually made in tobacco plants, which is interesting, but it also kind of limits the manufacturing capabilities of that particular therapy. So just moving on to talk about our drug, GS-5734. And so we first identified the parent drug of this molecule through a screening panel that we had performed at CDC. And so we saw pretty good EC50 values against Ebola across a number of different cell types. But our chemists at Gilead thought we could improve upon that. And so what they did was they modified the molecule. And so I'm not a chemist, so I'm just going to go with what our chemists have told us. And this part of the molecule that they added is supposed to help uptake into the cells. And then we have this first phosphate group on here because our drug is a nucleoside analog. And that first phosphate group is supposed to be like a rate limiting step for addition of subsequent phosphate groups to generate the nucleotide. So you can see that with the Prodrug now, we see much more potent activity in the multiple cell types with our drug. So I kind of alluded a little bit to the mechanism of action there. Once our drug is taken up into the cell, this group is cleaved off by esterases and cathepsin A. I think the enzyme which cleaves this group is really dependent on which cell type it's being taken up by. And that generates this monophosphate, which is then quickly converted into the triphosphate by the addition of these two phosphate groups. And so the mechanism of action of these nucleotide analogs is via inhibition of viral RNA polymerases. So then it's also important for us whenever we're looking at any of these new molecules is to make sure that there's selectivity over viral polymerases and not host polymerases. And that's what's shown here in this particular table. And then we wanted to make sure that when we're adding this prodrug to human macrophages that we're actually seeing generation of triphosphate. And so in the graph here, we're showing that when we add just the nucleoside, which is without the first phosphate group, you do see some level of triphosphate formation within the human macrophages. But when we have our prodrug, this much higher levels of triphosphate is detected in the human macrophages. So something that we're very interested in this molecule is the fact that it shows cross activity against multiple different viruses, particularly viruses that there are no approved therapies for, and they have the potential to cause widespread public health threats. So MERS and SARS, and Nipah hendra also. And then as you can see across multiple filovirus family members, we also see pretty potent activity. So I'm not sure how familiar people are in the audience with different animal models of Ebola infection. And so we've shown in vitro that we have nice potent antiviral activity. The next step is to then check in an animal model that we're actually seeing this activity in vivo. And so there are a few different models available. However, the rodent type models, you have to use adapted virus. You can't use wild type virus, whereas in the NHP model, you can use wild type virus. And also the pathogenesis of disease in NHPs is very similar to humans. So far to date, we've completed a number of different, studies in Rhesus, and all this work is in collaboration with USAMRIID in Maryland. There's very few BSL-4 labs in the country that can actually perform these studies. USAMRIID is one of them. And so the model that we've been doing most of our studies in is using KitQuit, which is an isolate from 1995, and we infect the monkeys with 1,000 PFU IM intramuscularly, and that's 100% lethal. So it's a pretty stringent model. The control animals die within 7 to 9 days of virus inoculation. And there's a very small window in which they start to show clinical disease signs, and then the animal actually succumbs to disease. That window's roughly 2 to 3 days. So in our first study, we treated the monkeys with a 3 mg per kg dose, delivered IV, a daily dose, because obviously in these types of studies, it's difficult to dose the animals multiple times throughout the day, and also for an actual product, you wouldn't really want to have to keep dosing patients. So a single daily dose is preferential. And we initiated treatment on either the day of infection or 2 days post-infection. And we saw some promising results from that study, up to 67% survival. So then we went into our second study, and so we thought that to maybe increase this survival rate, we'll start with a loading dose, a higher loading dose of 10 mg per kg, and then we'll give them 11 subsequent doses of a 3 mg per kg dose, and we call that the maintenance dose. So we have the 10 loading dose and a 3 maintenance dose. And we started this therapy either on day 2, day 3, or we also went in at the high dose for the whole study as kind of like a positive control. And so here I'm showing the survival rate. We monitor these animals out to day 28. In our more recent studies, we've been looking out up to day 42. But as you can imagine, these studies are pretty expensive, especially in the BSL-4 labs, and keeping the animals all the way out to day 42 can be difficult to do for all studies. So as we've seen previously in the model, the vehicle control treated animals, they died or succumbed to infection between day 7 and 9. Our group where we initiated treatment with the loading dose on day 2, we saw 33% survival, and then we saw 100% survival in the 2 groups where we initiated treatment on day 3, which was interesting. And then since this is a virology symposium, I should put obviously the viremia data here. And so this is the plasma viremia data. And then we can see that the peak of the viremia is normally between day 5 and 7. And then the control animals start to succumb to disease. And then in our groups where we had the highest dose, we saw the biggest reduction in viremia. And then in our 2 groups, we did see some nice significant reduction in viremia. However, we saw a significant difference between these groups in terms of the survival, which is kind of interesting. So then, because the NHPs can't obviously tell us how they're feeling, there's a scoring system and where the NHPs are given a clinical responsiveness score. And we can see here that in terms of disease signs, the 2 groups where we saw 100% survival, we saw a significant amelioration of clinical disease signs in those animals. And since hemorrhaging is kind of a hallmark of Ebola virus disease, we do perform pretty extensive clinical chemistry panels. And so I'm not showing you all of that data, but just in this slide, we thought I would show the fact that you don't see such a huge reduction in platelets in our treated animals. And so those studies I've shown you are being with using the Kikwit strain, which was an isolate from 1995 outbreak. We actually had access to the Makona strain, which was also a Zaire strain, but it was the kind of causative agent of the West African outbreak. So in this study, we actually just went with the highest dose group that we had used in the previous study. And it's a similar model where we infect the animals with 1,000 PFU intramuscularly. And we initiated therapy on day 3 post-infection. So in this particular study with this virus, the different Zaire virus, Makona instead of Kikwit, we did see a slight prolongation of survival in our vehicle control treated animals. However, they all did succumb to infection. And then we saw 100% survival in our treated animals. And then just to show the matching the viral RNA in the plasma, again, you see this peak around day 5 and day 7 of the viral load in the plasma in the control-treated animals. And then in our drug-treated animals, this is greatly reduced. So that's kind of a background to our non-clinical studies that we've completed so far. There are a few other studies ongoing, and we're also looking at efficacy against additional filoviruses, such as Marburg. But we have already completed a few clinical studies, and I thought it would be important today just to show you some of that. So this is kind of an interesting program at Gilead because most of Gilead's products already have been approved through the traditional approval pathway in which you collect efficacy data in patient populations. So that's interesting in the fact that it makes the process a little bit simpler, but in terms of something like Ebola, where there's not necessarily those thousands of patients available where you can actually perform these large-scale clinical trials, there is a pathway that is established at the FDA called the Animal Rule. It's a non-traditional licensure pathway. So when there's not going to be a current outbreak where we can actually test our therapy in patients, you can actually infer efficacy from an animal model, a very well characterized, validated animal model. And with that data and strong PK data and safety in people, you can actually achieve licensure of your drug. And so examples of those is Biothrax, which is a vaccine against anthrax, and Cyanokit, which is actually an antidote for cyanide poison. And both of those products were approved under this animal rule. And so although we've already completed phase 1 studies, safety and PK studies in people, we've also actually have 2 instances where our drug was used under a compassionate use basis for Ebola-infected patients. The first case was a female nurse, Pauline Caffrey. I don't know if that name rings a bell with anyone that's been following this, but so she was working in an Ebola treatment unit in Sierra Leone, was diagnosed with acute Ebola infection in December of 2014. She had very high viral loads, and she was treated with the ZMapp antibodies. And then she survived, and her viral load, her viremia disappeared. It was undetectable, and she was discharged the following January. However, a few months later, she relapsed, and they found high viremia in her CSF. And so then we treated her. We had the opportunity to treat her with our drug at different dose levels initially. And then we saw that even at this high dose level for 11 days, it was very well tolerated, and she cleared the virus from her CSF and has recovered subsequently. So our second patient, it was baby Nubia. She was born in one of the Ebola treatment units. Her mother did not survive the Ebola infection, but we treated baby Nubia with a smaller dose, obviously, of our drug, and it was tolerated very well, and she cleared infection and recovered, and will be celebrating her second birthday this year. So one thing that's interesting about the past outbreak is it's been estimated that there's approximately 17,000 survivors right now from that outbreak. And what has been shown is that Ebola can persist, especially in that nurse who we thought had cleared the infection and survived, but obviously the virus was persistent somewhere, most likely in the eye and the brain. So it has been shown that is the case. So in immune privileged sites, eye, brain, genital tract. And then here in this table, it was reported in male survivors that even out as far as 9 months, approximately a quarter of those survivors, you could still detect Ebola RNA in the semen samples. And it has also been documented that sexual transmission of Ebola has occurred. So then when we look at our tissue distribution studies that we did in the NHPs, we can see that in the male NHPs, we see actual distribution or good levels of our drug in some of these sites, so in the genital tract and in the eyes. So because we know our drug can penetrate those particular sites, in collaboration with NIAID, we have a phase 2 study that's currently ongoing, where we're collecting data from male survivors who have viral RNA persistence in the semen samples. And we're treating these men with 100 mg IV once daily, and then we're collecting samples to test to see if they can clear the RNA from those samples. And so we initiated this study on July last year. The study enrollment is still ongoing because obviously now it's getting harder and harder to find these patients. So we're looking forward to when we're going to be able to present some of that data. So with that, that's my last slide. Hopefully I've brought us back on track a little bit. And so obviously these studies have spanned from early in vitro testing all the way through to clinical testing. So there's been a number of people involved in these studies. And so I'd just like to thank all our collaborators at USAMRIID, the team at Gilead, CDC, Royal Free, MSF, who we worked with to treat baby Nubia. And for our funding for this project, for the non-clinical studies, came from the DoD at the US government. So with that, I'd just like to take any questions. Thanks.
So I think there's some Ebola going on now in the Congo. Are you guys, how do you physically react to these quickly enough to get your drug there? Or are you working on that? Or?
Yeah, so we're in conversations right now with MSF and NIAID because our drug is out there. It's in Liberia for phase 2 study, and NIAID has it there. I think right now, since it's a fairly contained outbreak, it's again in a very remote area where there's no roads really to get to that place. We think it's going to be one of those just small outbreaks. And I think right now, WHO and MSF are trying to gain approval from the Ministry of Health in the Congo to actually just kind of come in and start intervening potentially with the ring vaccination strategy is going to be their first point of call, yeah.
Actually, as a follow-up.
It's totally close.
As a follow-up, are you considering other routes other than IV, more practical routes like oral potentially, or maybe sub-Q?
And so for patients with acute Ebola, an oral drug probably wouldn't be absorbed quick enough to start the process of inhibiting the viral RNA. So that's why we think, and also when you think about these patients, gastrointestinal, vomiting, diarrhea, those are severe symptoms that these patients have. So whether they would be able to tolerate an oral drug is a question. So, and most of the time they have an IV line already.
Because you're probably thinking about this, it's not only the patients you want to benefit, but the healthcare workers also.
So yeah, so since there was good efficacy with the ring vaccination strategy, we think most likely healthcare workers, if they're just exposed but they don't necessarily have symptoms, they would probably get the vaccine rather than our drug. Our drug's mainly for the patient population that has acute viral disease.
I'd take both.
For the viruses that persist after treatment, are they in any way resistant to the original therapy?
That's a good question. So the nurse, they didn't give her the antibody again simply because they weren't sure if that was going to be able to penetrate the CSF. But when we've done viral resistance analysis in our NHP studies, we haven't seen any resistance in those studies.
Just to respond, I've seen also that your drug doesn't cross that well the CSF, right? When you presented.
Well, that was in the NHP study, yeah, and that was a single dose at a certain dose level. So we haven't done a dose response and looked at tissue distribution at higher doses. Okay. And it could be a timing thing as well. Yeah, that was just 2 time points.
Obviously, it works. So.
Yeah, exactly. Yeah, in the nurse case, it was very nice. We saw a steep drop-off as soon as we initiated therapy of the virus, Viremia and the CSF, yeah.
It's amazing as well, the distribution between all the negative strand RNA viruses. I mean, not all of them, but it's
and that's one of the things we're really excited about, right.
I understand.
Hi, just one quick question. It looked like in the treatment group initiated on day 2, there was less efficacy. So do you have any comment on why that might be compared to initiating on day 3?
Yeah, so we think that's related to the drug levels at the peak of the viremia. So as the viral replication is kind of taken off, when we initiate the loading dose on day 2 versus day 3, those animals on day 2 are going to have lower exposure of our drug than the animals where it's initiated on day 3 as the virus is starting to peak.